Okay, next topic, indicators of healthy outdoor environments. This one's where we really start to get into some links to some things we've done on trips and get a bit of a more hands-on into what we can do and what indicates whether some of our environments are healthy or not. So this is a topic that will link back even to some of the things we did on the first trip away. An indicator is anything that tells us what shows us how healthy the environment is or not. First one, there's 5 key ones. Water quality and adequacy. So quality self-explanatory, and adequacy means is there enough of it. Water quality, there's a number of things that influence what the quality of water is in an area. Environmental events, land use and human activities within the river systems. Sure, we've all seen some of these on some of the trips so far. Environmental events such as bushfires, floods, the two main ones, droughts even in a larger scale, land use, whether land's natural, whether it's being used for farming, other types of primary industries that can affect the water quality and adequacy, as we saw with gold mining in the Yakandander area all those years ago, and human activity as well, so towns and other towns and recreational activity that can affect water quality as well. Okay. How do we judge what the water quality is? There's a couple of ways. Nice and simply, how clear and clean it looks, and the type of organisms that live in it. So clean, clear water, generally flowing, but not always flowing, that has large diversity of organisms living in it, not just mosquitoes, but a whole heap of different things from fish to different types of water beetles. That would indicate that the quality of the water is quite good. Scientifically, we can test the turbidity, the pH, dissolved oxygen, things like that as well, to give us some measures and compare those against a database. Try and think of examples where we've done those or been able to do that on the 2 trips so far this year, or in other areas that you've visited or that you know about. Air quality and adequacy. Same thing, refers to the condition of the air relative to the requirements of one or more species or any human need or purpose. And there's some benchmarks for that. The EPA, the Environmental Protection Authority, has set some standards, and also to the levels of different pollutants or different chemicals that are allowed in the air. Things like carbon monoxide, nitrogen dioxide, ozone, and other things, other chemicals that are there that may indicate that the air is not that healthy. We'll have a bit of a look at that in class, because we can actually look at some of the Victorian air monitoring stations and start to get a picture of what the air quality is like at different parts of Victoria in real time. There's an example of Box Hill, which is in Melbourne, air quality measurement. Right, soil quality and adequacy. The way we measure this is things like vegetation cover, the health of the vegetation, diversity of vegetation too, whether not just that there's vegetation cover, but is there a variety of vegetation cover there. Salinity levels, salty soil will only allow or will only support salt-tolerant plants, which are very hardy but don't indicate that the soil is very healthy. Things like land degradation, compaction, erosion, nutrient levels. Some really simple thing with the compaction. Soil, for plants to grow and species to grow in the soil, it needs to be quite friable. The term friable means that it crumbles. And so if it's compact and really tightly compacted, the roots of the plants can't penetrate it, and so you don't get a good variety of species going. So a really simple test is to actually take some of the soil in an area and crumble it. Is it sandy? Is it clay? Is it crumbly? And also from that, is it wet and gluggy? If it's not draining well, even if the other things are right and it's really gluggy, then that means the soil's not that healthy. And by checking a sample of soil, you also look at what other things, if there's bugs like worms and little beetles and microorganisms in there, that indicates healthy soil as well. And then we can do things like testing soil pH and soil nutrients and things like that in the lab or in the field with a few simple tests as well to measure soil quality and adequacy. The adequacy part, you look at topsoil being washed away, lots of erosion, that would probably indicate that not good adequacy of soil, so that indicator is not that healthy. Levels of biodiversity. We'll delve a little bit deeper into biodiversity, but biodiversity is the diversity of life in an area, and it works on 3 levels: genetic biodiversity, species biodiversity, and ecosystem diversity. The amount of genes simply means the number of different individuals within a species, because you have greater genetic diversity if there's thousands of a certain type of bird species or a certain type of plant species in an area. The same with the number of different species within an area. A great example of that was at Stanley, where on one side of the road was the pine plantation, on the other side the native forest. The species diversity on the pine plantation side was pretty poor because there was only one species in there, or predominantly one species. On the other side, the species diversity was pretty good. Genetic diversity might have been pretty good in the pine plantation because we had a huge number of individual trees of that same species, but species diversity not so good. And then ecosystem diversity, the amount of different ecosystems in an area. So do we have some dry forest woodlands, some rainforests, some farm grasslands, a mixture of different ecosystems. Generally this refers to more, you'd look at this more on a statewide basis rather than just a local area basis. The higher the levels of those, then there's of each of those, then the healthier the environment is. And what is biodiversity? A little bit of a backtrack here. Just read that. And yes, in the study design, you do need to be able to know and assess the levels of each of the levels of diversity within an area. We'll watch this video now.
I'm an adjunct professor of biology, and today we're going to talk about biodiversity. First, let's start by breaking up the word. Now, the first part, bio, that means life. The second part, diversity, that means differences. So think about the diversity in humans, how we're all very different. Now, if you put those two words together, you're talking about the differences of life. Now, high biodiversity, or having lots of biodiversity, is always a good thing. Let's talk about the 3 different types of biodiversity. We're going to go from smallest to largest. First, genetic diversity. Genetic diversity is important within a species or a population because that's going to give that species the greatest chance of survival. When you have high genetic diversity, you're just going to have more genes in that gene pool available to deal with environmental changes. Now let's move to the middle one, species diversity. Now species diversity is going to be the different types of species you have in an ecosystem. However, if you're talking about, let's say, a tropical rainforest, you're going to have huge, high species diversity. That means that ecosystem is doing well. But even if you talk about a desert, that diversity is going to be a little bit lower, but you still want high species diversity that's relative to that desert. And lastly, our largest type of biodiversity is ecosystem diversity. On the planet, you want lots of different types of ecosystems. You don't want just one ecosystem, let's say all grassland. That would only support grassland species. So by having lots of different types of ecosystems, you can support lots of different types of organisms. On our planet, we have so many different types of ecosystems. Tropical rainforests, we have deciduous forests, we have tundra, and all of those different types of ecosystems help to make sure that our planet stays nice and healthy. Thanks for watching, and if you want to learn more about this subject, click on the link below. Or if you want to learn more biology, feel free to click on any of the links around me. And please, rate, comment, and subscribe to this channel.
So back to where we were before, we need to be able to assess the levels of biodiversity. We can do that by simple things like just doing a species count, like we count the number of plant species in an area, in a small area, and just look around to see. You don't even need to name them, just to be able to identify that they are different is a good way to do species diversity.
The woodlands and shrublands of Central Australia and the small pockets of forest that we have along the east coast and the far southwest of Western Australia. The climate, the rainfall, floods, droughts, fires, and things like that all work together to create an extremely diverse set of environmental conditions that promote diversity within Australia.
It can be really amazing, the biodiversity you find in really good habitat, and that could be anything from grasslands to forests. It was really brought home to me when I did a quadrat survey. Now, a quadrat survey involves throwing a square metre frame down on the ground. And when I looked in this frame in the forest behind my place, I was finding between 20 and 30 different species of plants in that square metre. It's up to 50 different plants per square metre. So it was an amazing discovery for me anyway.
So Australia is filled with biodiversity, more than most places. But it's a pretty tough place to live in. It's a very dry country, and the ecosystems can be pretty fragile. It doesn't take much to bring on severe environmental damage.
Right, and the last indicator of how healthy an outdoor environment is the levels of introduced species or pests, and that, I guess, is pretty self-explanatory. By looking at the numbers and types of introduced species in an area, can tell how healthy it is, because more introduced species will compete with native species for habitat. Weeds like the blackberry will choke out around creek beds, choke out native grasses and water bank, creek bank species. Foxes, rabbits compete with native species of animals for food. Rabbits damage native plants, and therefore that throws the ecosystem out of balance at all levels, and can mean that the environment's unhealthy. I guess an indicator of that, of introduced species, the people that went on the Kawombat Flat bushwalk, the impact of the brumbies would mean that in some areas you could see the damage and the lack of health to the environment caused by the introduced species, by the brumbies. What you need to be able to do in this outcome then is, for each of the measurements of health, you need to be able to assess that health across those areas for a variety of environments. So we've started to do that now, even way back in term one, on the first trip, we looked at some factors that it would indicate how healthy those couple of environments we went to were from. And start to build up a picture of the health of the different environments. So do they make the environment healthy or not? What evidence do you have to support that factor in the environment, the health? So what evidence have you been able to use from an environment that you visited or studied to measure the health of the quality and adequacy of water or soil or air or the levels of biodiversity, or the levels of introduced species. Be able to discuss what the indicators tell us with reference to your trip.